natural plasmid vector pSC101. P stands for plasmid, SC stands for the discoverer of the plasmid. SC is a person who discovered this plasmid. He is Stanley Norman Cohen, and 101 is the number of plasmid which is identified in the same laboratory. So today's topic we have is plasmid vectors, natural plasmid vector pSC101. Can you see? Can you see? Give me some voice.
Yes, ma'am.
Okay. So plasmid vectors, natural plasmid vector pSC101. pSC101 is a resistant plasmid, natural resistant plasmid, or what you simply designated as R plasmid. Stanley Norman Cohen, who was working in Stanford University, identified pSC101 in the year 1972, which is a resistant plasmid. So Stanley Norman Cohen, who's also written as Stanley N. Cohen or Stanley Cohen. So Stanley Norman Cohen, who was working in Stanford University, identified pSC101 in the year 1972, which is a resistant plasmid. It is a natural plasmid of Salmonella Panama. So the organism that possesses this plasmid is Salmonella Panama, an antibiotic resistant plasmid which is about 8% of the original DNA. So it is a natural plasmid of Salmonella Panama, an antibiotic resistant plasmid which is about 8% of original DNA. Size of the plasmid, if you see, is around 5.8 M Daltons and nucleotides that approximates to 9,000 base pair. Exactly, it is 9263 base pair or 9.2 kilo base. So regarding the size of the plasmid is around 9.2 kilo base in size. pSC101 has, or it possesses resistance to the antibiotic tetracycline. So if you look into the figure on the right hand side bottom, you have an pSC101 vector system that has an tetracycline resistant gene. It has an origin of replication to autonomously replicate. The size of the plasmid is mentioned in the center and one restriction site is mentioned over the vector, that is EcoR1. So pSC101, the plasmid vector, shows to the resistance of the antibiotic or for the antibiotic tetracycline. If any cell possesses this plasmid, for example, Salmonella Panama produces this plasmid naturally. In case if this plasmid is present in the organism will remain resistant to the antibiotic tetracycline. Various restriction sites present in the plasmid includes ZO1, EcoR1, PVU2, HING2, BAM-H1, SAL1 and HIND3. So various restriction sites that are present in the vector pSC101 is ZO1, EcoR1, PVU2, HING2, BAM-H1, SAL1 and HIND3. If you can use any of these, you can cut what is called as pSC101. That is why you call them as multiple cloning or polylinking site, because you can restrict it with so many different restriction endonuclease, and this vector system possesses unique restriction site for so many different restriction endonucleases. So just looking at the picture now, pSC101, the size is around 9,000 base pair approximately. It is appropriately, if you see, it is 9.2 kilo base. It has resistance to the antibiotic tetracycline. It has an origin of replication to autonomously replicate, and restriction site which is present includes ZO1, EcoR1, PVU2, HING2, BAM-H1, SAL1 and HIND3. So I will go again with the slide once. Stanley Norman Cohen, who was working in Stanford University, identified pSC101 in the year 1972, which is a resistant plasmid. It is a natural plasmid of Salmonella Panama, an antibiotic resistant plasmid, which is about 8% of original DNA. Size of the plasmid is about 5.8 Daltons, and nucleotides approximates to 9,000 base pair, which is 9.2 kilobase. pSC101 possess resistancy to the antibiotic tetracycline. If you look into the bracket, it is given TET-R, which means it is tetracycline resistant. In case if any antibiotic is sensitive, it will be mentioned as TET is yes or AMP yes. Whatever antibiotic shows sensitivity, it will be mentioned as yes. If it is resistant, it will be designated as R. So TET-R gene it possesses, and various restriction site present in the plasmid include ZO1, EcoR1, PVU2, HING2, BAMH1, SAL1, and of course HINT3. pSC101 is the first vector used for gene cloning experiment possessing tetracycline gene as selectable marker. I repeat the sentence. pSC101 is the first vector used for gene cloning experiment possessing tetracycline gene as selectable marker. What does this word mean? Selectable marker. Look into the picture now with concentration. You have a plasmid on the left side of the diagram, top left, topmost left. You have one restriction site which is mentioned of EcoR1, and gene for antibiotic resistancy is colored green. On the right hand side, if you see, you have a foreign DNA fragment which is mentioned slightly purplish blue with your region of interest, that is DNA of interest, which again has restriction site for the restriction endonuclease EcoR1. I have already told you the restriction endonuclease that is used to clip the vector molecule is equivalently used to clip the foreign DNA fragment. You cannot change the restriction endonuclease to cut foreign gene and the vector. You should use the same restriction endonuclease to nick the foreign gene as well as the vector molecule. So plasmid here on the top left has gene for antibiotic resistant with EcoR1 restriction site. On the other hand, on the right, you have a foreign DNA fragment with the restriction endonuclease site EcoR1 with a region of interest which is colored pink. Okay, so you are now restricting it with what is called as restriction endonuclease type 2 EcoR1 to produce a sticky ended molecules. Once you produce a sticky ended molecules, you hybridize it with the help of enzyme DNA ligase. I already have mentioned you DNA ligase is an enzyme that forms phosphodiester bond, or it joins DNA fragments and thereby producing a recombinant DNA molecule or chimeric DNA molecule. So the third stage of the figure, if you see, you will have the plasmid with a combined foreign DNA fragment of your interest. Now the next procedure is you have to select the cell after transforming it into the host cell so that the cell has taken up the recombinant molecule. So the recombinant DNA is inserted inside a bacterial cell with the help of phenomenon called as transformation or transfection, whatever you mention it. So you're transforming the plasmid DNA, that is chimeric molecule, inside the bacterial cell. Come down of the plasmid left-hand side figure out. You can see you have a bacterial cell. It has bacterial chromosomal DNA and the inserted plasmid vector, which is black in color. Now what will the bacterial cell do is it will multiply by binary fission, developing the clone. Now the next procedure is you have to identify this clone. So what you will do is you will take a bacterial plate that is incorporated with antibiotics. So the medium is incorporated with antibiotic. For example, in case if you're going to screen the organism containing pSC101, you will have a culture media in which tetracycline is incorporated. So I already told you the organism that possesses this plasmid will resist tetracycline, so it will grow in what is called as the plate incorporated with tetracycline molecule. Since the antibiotic resistant gene is used to select the recombinant molecule, it is called as selectable marker. Hope it is clear to you all. So pSC101 is the first vector system used for gene cloning experiment possessing tetracycline gene as selectable marker. In the particular gene vechu நம்ம recombinant cell select பண்ணக்கூடியனால you call this as selectable marker. So first prokaryotic plasmid in which eukaryotic gene from an African toad Xenopus lewis rRNA was cloned. So this vector system was first used in gene cloning, and they experimentally cloned eukaryotic gene rRNA of an African toad Xenopus lewis in an prokaryotic plasmid pSC101, and the experiment was carried out by Herbert Boyer, Stanley N. Cohen, and Paul Berg. So that is why they are referred to as fathers of rDNA technology. So Herbert Boyer and Stanley Norman Cohen, they played a significant role in recombinant DNA technology by initiating cloning for the first time. So they are designated fathers of recombinant DNA technology. And of course, the experimental proof was brought out by Morrow et al. Proved the above cloning of a eukaryotic gene in the year 1974. I repeat the slide. First prokaryotic plasmid pSC101 in which eukaryotic gene from Xenopus lewis African toad rRNA was cloned. It was carried out by Herbert Boyer, Stanley Norman Cohen, and Paul Berg. Morrow et al. Proved the above cloning of a eukaryotic gene into a prokaryote in the year 1974. What was the experiment done? Just look into the picture below. So they took the vector pSC101, which shows resistance to the antibiotic tetracycline, and one site of restriction endonuclease is mentioned, EcoR1. They took they isolated the rRNA genome of African toad Xenopus lewis, and both these were restricted with the restriction endonuclease EcoR1. So look into the left-hand side pink color, you see pSC101 vector. You can see the purplish one, the genome of Xenopus lewis, the African toad. It is restricted or it is cut with the restriction endonuclease EcoR1, and it is mixed or ligated with the enzyme ligase to produce an chimeric molecule. So come to the down portion, you can see pSC101, the chimeric molecule, which is pink and purple joining together. So gene for rRNA. You are transforming or introducing it into the host cell. The host cell commonly used here is Escherichia coli. So whenever you call the term host cell, you should remember two things in mind. It should be Rec A minus. So Rec A minus, which means unnecessary recombination should not take place between the chromosomal DNA and plasmid. So the strain should be recombinant A minus. And of course, the strain also should be RE minus, restriction endonuclease minus, because if it produces restriction endonuclease, it will cut the vector again once it enters the cell. So the cell that is used as a host strain in gene cloning experiment should always be Rec A minus and RE minus. That you need to remember in your brain when you're going for further classes. So it is transformed to Escherichia coli and first genetically engineered plasmid introduced to E. coli vector. And of course, the vector can autonomously, the plasmid can autonomously replicate because it has origin of replication by producing multi-copies of recombined gene. So it is identified in an plate containing tetracycline. That is why it is called a selectable marker. Tetracycline resistant gene is called a selectable marker. So the recombinant cells are identified using the plate containing the antibiotic tetracycline. So initially what was carried out was cloning an eukaryotic vector system or eukaryotic foreign gene inside an prokaryotic vector system. And later on, it was also found that a prokaryotic foreign DNA can be isolated and can be cloned in an prokaryotic vector molecule. So two prokaryotes can be joined together. So expression of Staphylococcus plasmid genes into Escherichia coli. So before going into the slide, we will pass on to the next slide. Okay. So what was the experiment all about, or how did prokaryotic genes were connected to each other, or how was these cloning done? If you see, two different plasmid was taken for the experiment. One is pSC101 of the organism Salmonella Panama, and the other plasmid that was used for the experiment is P1258, the plasmid found in the organism Staphylococcus aureus. So two different plasmid was taken for the experiment. One is pSC101, the other one we have is Staphylococcus aureus plasmid P1258. P1258 is a plasmid that showed resistance to erythromycin, mercury, cadmium, and ampicillin. So if you look into the picture, you can very easily understand. It shows ERY resistance, Hg resistance, cadmium resistance, and ampicillin resistance. And these can be cleaved with the restriction endonuclease into 4 different identifiable fragments by using the restriction endonuclease EcoR1. If you see EcoR1, it is in 4 different places in the picture. So if you use EcoR1 and cleave this molecule, you will get 4 different fragments. One is for erythromycin resistancy, the other one is for mercury resistancy, one is for cadmium resistancy, the other one is for ampicillin resistancy. So what was the experiment? What was the plasmid taken for cloning prokaryotic genus? P1258 that showed resistance to 4 different molecules, the erythromycin, ampicillin, mercury, and of course cadmium, and it can be identifiably cut into 4 different fragments with the help of restriction endonuclease EcoR1. Now go into the right side of your picture. Okay, so you have 2 different diagrams here on the top. pSC101, which has tetracycline resistant G and can be restricted with the enzyme EcoR1. On the other hand, with purple color, P1258 that has 4 different resistances, erythromycin, ampicillin, mercury, and cadmium, that also can be restricted with the restriction endonuclease EcoR1 into 4 different fragments. So you're using restriction endonuclease EcoR1, you are nicking the molecule. If you see the second stage of the diagram, so if you come beneath, you can see an opened pSC101 and a 4 fragmented P1258 molecule. These 2 you are ligating with the help of the enzyme ligase, so that you find out beneath that P1258 plus pSC101, which is a chimeric molecule or a chimeric DNA that shows a resistance to ampicillin, tetracycline, cadmium, mercury, and erythromycin, and this molecule is transformed to the host Escherichia coli cell. Now the cell which possesses the plasmid will show antibiotic resistance to 2 different or 3 different antibiotics, that is erythromycin, ampicillin, and tetracycline, which means the combination has occurred, and checking of the transformants can be carried out with a medium incorporated with ampicillin and tetracycline. If it is only tetracycline, it is only pSC101. If it is ampicillin and tetracycline, it is of course a chimeric molecule that is present in the cell, which has undergone what is called as combination or recombination or recombined molecule. That is what is the experiment all about. So just look into the picture again. pSC101 and P1258 restricted with restriction endonuclease EcoR1. Here on the left hand side, pSC101 1 fragment, on the other hand P1258 4 fragments. You are ligating it with the enzyme ligase to produce a chimeric molecule, transforming it into a host Escherichia coli cell. Now the Escherichia coli cell that has got the recombined or chimeric molecule will show resistance to tetracycline, ampicillin, erythromycin, mercury, and cadmium, and of course it is checked with the medium containing ampicillin and tetracycline. Now this plasmid will have ampicillin and tetracycline as selectable markers, because now they are using 2 different antibiotics to select the recombinant cell. Now we will go to the previous slide so that you can understand. So expression of Staphylococcus plasmid genes in Escherichia coli. So 2 persons who played an important role is Chang and Cohen in the year 1974, who considered Staphylococcus aureus plasmid P1258 as being particularly appropriate for the experiments involving interspecies genome construction. So two different species of prokaryote, they are constructing together. So interspecies genome construction, since it carries several different genetic determinants that were potentially detectable in Escherichia coli. So I repeat the sentence. For this experiment, Chan and Cohen in the year 1974 considered Staphylococcus aureus plasmid P1258 as being particularly appropriate for the experiments involving interspecies genome construction, since it carries several different genetic determinants that were potentially detectable in Escherichia coli. Moreover, agarose gel electrophoresis indicated that this plasmid is cleaved by the EcoR1 restriction endonuclease into 4 easily identifiable fragments. Molecular chimeras containing DNA derived from Staphylococcus and Escherichia coli were constructed by ligating a mixture of EcoR1 cleaved pSC101 and P1258 DNA, and then were used to transform a restriction-less strain of Escherichia coli. That is what I meant, RE minus. If this host strain produces restriction endonuclease EcoR1, then pSC101 and P1258 gene that is cloned will be cleaved. That is why the restriction-less strain of Escherichia coli has to be used. E. coli transformants that express the ampicillin-resistant determinants carried by Staphylococcus plasmids were selected and checked for tetracycline resistancy. So I repeat this slide. For this experiment, Chan and Cohen in 1974 considered Staphylococcus aureus plasmid 1258 as being particularly appropriate for the experiments involving interspecies genome construction, since it carries several different genetic determinants that were potentially detectable in Escherichia coli. Moreover, agarose gel electrophoresis indicated that this plasmid is cleaved by the EcoR1 restriction endonuclease into 4 identifiable fragments. Molecular chimeras containing DNA derived from Staphylococcus and Escherichia coli were constructed by ligating a mixture of EcoR1 cleaved pSC101 and P1258 DNA, and then were used to transform a restriction-less strain of Escherichia coli. E. coli transformants that express the ampicillin-resistant determinants carried by the Staphylococcus plasmids were selected, and then it is checked with or checked for tetracycline resistant gene. So that is the reason ampicillin and tetracycline in this case is selectable markers. So now you can very easily understand this diagram. For the better understanding, this is a flow chart for what is happening with the prokaryotic genome construction. So pSC101 has tetracycline resistant gene, P1258, 4 different resistance Z, ampicillin, erythromycin, mercury and cadmium. EcoR1 restriction endonuclease is used to cleave. Now go into the left hand most. So pSC101, 1 fragment. If you ligate, if you transform to Escherichia coli and select on ampicillin, it does not have an ampicillin resistant gene, it has only tetracycline, so transformants will not be grown. So self-ligate item, it will not grow. On the other hand, go to the right hand extreme. Okay, P1258, 4 fragments, ligate, transform. Oh, so no tetracycline transformants are formed. So there is a spelling error, of course. There will be ampicillin resistancy, but no tetracycline resistancy will be there. Now come into the center portion of your diagram. So you are mixing 1 fragment with 4 fragments, ligating. The transformants selected in the ampicillin plate. So P1258 ampicillin gene, PSC could join panirkom. So it will show ampicillin resistancy, it will grow. Then you will take the colony, check for tetracycline resistance. Now the recombinant cell will show the ampicillin and tetracycline clones containing plasmid chimeras. So that is what is the